Welcome back to my teacher program. Now let's see a small video of cell cycle. Hope you enjoyed the video. So hope this is clear to you, the cell division. Now we'll move on to the next type of cell division, that is the meiotic cell division, meiosis. Now this meiotic cell division, remember that this takes place in germ cells. So what is the significance of this meiotic cell division? This is, we have seen that meiotic cell division helps to reduce the chromosome number to half. So we can see that if an organism or a cell has got 23 pairs of chromosomes, 23 pairs of chromosomes, after meiotic cell division, total 4 cells are produced and each cell has a haploid set of chromosomes. That is only 23 number of chromosomes. So we have seen that here the cell starts with a cell with 23 pairs of chromosome. Now the cell ends with 23 number of chromosomes. So what is the significance? As I think I have mentioned earlier classes, that when the gamete or the fusion of gametes takes place, again we can see that spermatozoa with the 23 chromosomes fuse with the ovum with the 23 chromosomes, and a somatic cell is produced or a zygote is produced. Zygote will be having 23 pairs of chromosomes. Now to retain the number of chromosomes, it is very essential the cell has to undergo the meiotic cell division. Now we can see what are the various stages of meiotic cell division. Meiotic cell division also, there are 2 meiotic phases. We can call it as meiotic 1 and meiotic 2 or meiosis 2. Meiosis 1 is a large phase. We can see that this is again divided into different phases. As we have discussed, it is again divided into prophase, metaphase, anaphase and telophase. So again these are the same phases which we have studied earlier. Now in mitosis, prophase, metaphase, anaphase and telophase. But remember that meiotic cell division, the prophase is again, or we can say that to avoid confusion, we can call it as prophase 1, metaphase 1, anaphase 1, then telophase 1. The prophase 1 is again divided into various phases. These phases are leptothene. Some books you can see the leptonema, both are correct. Leptothene, zygotene, pachytene, diplothene and diakinesis. So we can see that this prophase 1 is again divided into 5 phases, 5 stages: lactotin, zygotin, packaging, diplotin and dikinesis. So what are the changes taking place during prophase 1 of meiotic cell division? Here the prophase 1, we can see that the cell, it is a, as we have discussed, cell is the normal cell which is having a nuclear membrane and nuclear materials or the chromatin fibers are present. Then nucleolus is present. As we have discussed, we can see that first the chromosomes will appear, or the condensation of chromatin materials taking place. When the condensation of chromatin material taking place, we can see that it attains a shape of a chromosome. When these chromosomes are formed, you can see that there are a set of chromosomes which we have obtained from our father and a set of chromosome which is obtained from mother, or a zygote has got a pair of chromosome, as I have mentioned. One set is from the mother and one set is from the father. So these similar chromosomes, we call it as homologous chromosomes. So we can see that these homologous chromosomes, first the doubling of the chromosome taking place, chromosomes will increase in number. Then in the later stage of prophase 1, we can see that homologous chromosomes come close together. That is, you can see that here the homologous chromosomes are coming close together. I am just showing one as a larger one and one as a smaller one to identify the chromosomes. Here we can see the homologous chromosomes close together. Now what is happening here? Now this is, we can see this is what happening in the case of zygotin. What is happening in packaging? A chiasmata formation, a crossing over taking place. That is, we have seen that the genetic material of one chromosome will get exchanged with the genetic material of the other chromosome. This process of exchange of genetic material is known as crossing over. Now let us see what is the importance of this crossing over. Crossing over is very important because which helps to generate variation in cells. That is, we have seen that a human being or an animal can produce different types of gametes, in the sense it has different sets of genes. This helps for the variation, and you have studied, might have studied in evolution, variation is essential for speciation. That means the new species formation. Now let us concentrate on this. We can see that the crossing over taking place. Remember that the crossing over takes place in the same locus. Same locus, same position. Suppose here, if this part is broken and attached, only the same locus, the crossing over taking place. Now also remember that crossing over always takes place between the non-sister chromatids of the homologous chromosome. Normally students are getting confused, what is a non-sister homologous chromatids? When we are having a chromosome with 4 chromatids, these 2 chromatids we can call it as sister chromatids. These are the sister chromatids. When we are having a homologous chromosome, we can see that this and this, these 2 chromatids are known as non-sister chromatids. Non-sister chromatids of homologous chromosome. So crossing over taking place between the non-sister chromatids of the homologous chromosomes. Then later, after the non-sister crossing over, chiasmata disappears and homologous chromosomes set arranging in the equatorial plane. That's the end of the dikinesis phase. Now lactotin, zygotin, packaging, diplotin and dikinesis. In this packaging phase is comparatively larger phases compared to lactotin and zygotin, where the what is taking place? Crossing over taking place. Crossing over is very important, which is needed for the recombination of gametes. Recombinant DNA will be formed as a result of crossing over. Now what is happening here is the cell will undergo the metaphase 1. Let's see what is the difference between this metaphase 1 of meiotic cell division and metaphase of mitosis. Even in the mitosis, you have studied chromosomes arranged in the equatorial plane and centriole breaks, spindle formation taking place. But here also same thing, but we can see that homologous chromosomes are arranged. So remember that here centromere breaks and the chromatids are separated in case of anaphase of mitosis, but here in metaphase 1 we can see that homologous chromosomes are arranged in the equatorial plane. Then in anaphase, what is getting separated? Homologous chromosomes are getting separated. So we can see that you have studied now when the tetrad or the Pachytene stage crossing over. After the crossing over, homologous chromosomes will be arranged in the equatorial plane and these homologous chromosomes are getting separated. This question is always asking what is the difference between a metaphase 1 and metaphase 2, or you may expect the question metaphase 1 of meiotic cell division and metaphase of mitotic cell division. Hope this is clear to you. In metaphase 1 here what is happening is the homologous chromosomes are getting separated. Now we can see that as a result the meiotic cell division, first meiotic cell division is known as a reduction division, because number of chromosome, ultimately speaking we can see that the number of chromosome will be reduced. You can see here then in the telophase what is happening, a cell plate formation, and in the case of animal cell as I have mentioned, a conversion will be formed and the cell membrane will move inside, then a cell will be divided into two cells. Now here instead of here what is happening is the cell will undergo, after the telophase 1 the cell will undergo the second meiotic cell division. Now hope this is clear to you. Prophase 1, metaphase 1, anaphase 1 and telophase 1 and various steps of the prophase 1. Remember the Pachytene stage and the crossing over and the significance of crossing over. Then after the crossing over what is happening to metaphase 1 and anaphase 1. Hope this part is clear to you, because this you should know to understand the non-disjunction. You might have studied Down syndrome and different syndromes. In genetic chapter we have discussed about different syndromes, and these syndromes, the main reason for these syndromes are the increase in number of chromosomes. For example, you have studied in the case of Mongolism or Down syndrome, what is happening is the triploid condition of the trisomic condition of the 21st chromosome. That is, in the 21st chromosome instead of one pair of chromosome, what will be there? 3 chromosomes are present in Down syndrome, trisomic condition. So you may surprise how 3 chromosomes are formed. Normally it is one pair of chromosome. Here what is happening is 3 chromosomes are formed. This is due to non-disjunction of homologous chromosomes. Now let us see what is non-disjunction. This is related to your genetic topic, but you should know what is non-disjunction. Non-disjunction of homologous chromosomes. Here we can see that, as I have mentioned, when the anaphase 1 takes place, homologous chromosomes separated, but instead of separating the homologous chromosomes, both the homologous chromosomes together will move into a single cell. Here you can see that when this non-disjunction happened, what is happening here? Both the homologous chromosomes have gone to one cell, and this is the pair of this homologous chromosome. So ultimately speaking, here we can see that this cell has got a more number of chromosome, and this cell is lacking one chromosome. So this phenomenon is known as non-disjunction. As a result, what is happening is trisomy condition, or even in the case of Turner syndrome. Turner syndrome we have studied X0 condition. X0 condition means only one X chromosome will be there. There is no pair, either the X or the Y chromosome. So that is the reason for Turner syndrome. Even in the Klinefelter syndrome also we can see that either it become XXX or it is XXY. These all syndromes, diseases are due to non-disjunction of homologous chromosome. So remember that this meiotic cell division is very important. Now let us see the various stages once again. I will show you this. Various stages of meiotic cell division. Prophase 1, metaphase 1, anaphase 1, telophase 1. Hope this part is clear to you. Prophase 1, metaphase 1, anaphase 1 and telophase 1. Then the cell will undergo the second meiotic phase, that is prophase 2, metaphase 2 and anaphase 2. So now let us see what are the various steps of meiotic cell division 2. Meiotic cell division 2 is very easy to remember because this is exactly same like mitotic cell division. Again we can see the various steps of meiotic cell division 2, that is a small prophase will be there, then metaphase 2, anaphase 2 and telophase 2. So we can see, you can remember this very easily because these are the same steps, similar process what you have studied just now in mitosis, the same thing happening in meiotic 2. Once again I will repeat, here we can see the what is happening, centromere breaks in anaphase 2. So the question can be, what is the difference? Can anybody say the answer? What is the difference between anaphase 1 and anaphase 2? You can send us the answer as SMS or you can call us and tell me what is the main difference between anaphase 1 and anaphase 2. Anybody? Anaphase 1 and anaphase 2. The difference, it is in the case of anaphase 1, homologous chromosomes are separated. Homologous chromosomes separated. But here in anaphase 2, what is happening? Centromere breaks and chromatids are separated. Chromatids are separated. The rest of the process is same like mitosis. So you can see that ultimately after the cell division, 4 cells are produced. Each cell is having a haploid set of chromosomes. Hope this process is clear to you, and you should know the difference between mitosis and meiosis, and what is the difference between mitosis which is taking place in a plant cell and an animal cell, and what are the various steps of meiotic cell division. Because to understand the phenomenon or the theory of independent assortment of genetic theory, you should know meiotic cell division. Now I will show you once again the slides showing the various steps. Here you can see the crossing over process between non-sister chromatids, chiasmata formation, non-sister chromatids, chiasmata site of crossing over. See the overlapping and a tetrad formation. A tetrad means a centromere with 4 chromatids. Then comes to the sex chromosomes. XX and XY chromosomes. Y chromosome is comparatively smaller. metaphase 1, variation. It leads to the variation, and formula to find out, that is 2 raised to n. Now see this question. In terms of independent assortment, how many different combinations of sperm could a human male produce? In terms of independent assortment, how many different combinations of sperm could a human male produce? Can anybody give the answer? Please send us the answer, or you can also SMS. Based on the independent assortment, how many different combinations of sperm could a human male produce? The formula you have to use, 2 raised to n. Human chromosome 2n is equal to 46, n is equal to 23. 2 raised to 23 is equal to 8 million combinations. So you may wonder, a human male can produce 8 million combinations or different types of genetic materials in the spermatozoa. 8 million different combinations. Now let us see the anaphase 1. Homologous chromosome separated. Telophase 1. Each pole has now haploid set of chromosomes, then followed by cytokinesis. As I have mentioned, cytokinesis is the division of cytoplasm. Telophase 1, 2 cells are formed. Each cell has got half the number of chromosomes. Meiotic 2 or meiosis 2, no interphase 2. Same like in mitosis. Here you can see the difference between metaphase 1 and metaphase 2. Sister chromatids are separated and chromatid starts moves to the poles. Then comes to telophase 2, nuclei formation, and again the cytokinesis taking place. Remember, 4 haploid daughter cells are produced. Each cell is having half the number of chromosomes. So gametes is equal to either the sperm or the egg cell. This is how the fusion of gametes taking place. Now please answer this question. What are the 3 sexual sources of genetic variation? This question is very important. What are the 3 sexual sources of genetic variation? Point 1, crossing over. It takes place in the prophase 1, Brachytene stage. And second answer, second point, independent assortment which is happening in metaphase 1 and random fertilization. Hope this part is clear to you. What are the 3 major sources of variation? One point is the crossing over, independent assortment and random fertilization of gametes. Hope this is clear to you, and we will come back with another topic in the next class. If you are in between, if you have any doubt, you can contact us or you can SMS us and we will clarify your doubts. Wishing you all the very best, because as you know that you all are going to face the board examination and some schools already the model examinations are going on. Wish you good luck.